Hi, it's Mrs. Vance again, and this time we're taking notes, talking about evidence of evolution. So Darwin spent 5 years on this trip, saw lots of different things all over the world, observed lots of different things about geologic formations and geologic processes, as well as collected lots of specimens for the British Museum and cataloged them. And all the while he was doing this, he was making observations of his own, which later on informed his ideas about how life changed over time. And so we're going to look today at some of the evidence of evolution. This shouldn't be a real long video because you have an extensive worksheet that goes over this that you're going to spend a couple of days working on. So we'll just go through the basics so that you can see what kinds of evidences we're looking at and then draw some conclusions from that. So there were four main categories of evidence, okay? And we're going to talk a little bit in turn about each one of these. And so one is fossils. Darwin saw lots of fossils, and we still see fossils today that are very similar to modern species, but there's some differences there. And so what are the differences and how has that changed things over time? Secondly is biogeography. This is the locations of related groups in different parts of the world that have more to do with what the environment was like there at the time than what it's like now. The third point is homology. These are similarities in organisms that come from having had common ancestors, and there are quite a few things involved in this. Darwin called this common descent, meaning that we descended from ancestors that were similar or the same. And then the final one is something called vestigial structures. These are structures that are still present, but that are no longer used, and so they may have become reduced in size. So first of all, fossils. Now, oftentimes, fossils are found in geological layers of rock, okay? There are many fossils that resemble modern species. When you're looking at fossils that are embedded in rock, remember the deeper you go down in the rock, those are older fossils. The ones that are shallower, closer to the surface, are more recent. And generally what you find is the older ones are simpler and the more recent ones are more complex. Now, there are lots of ways to determine the age of the rock. Some of it has to do with radioactive isotopes and how quickly they decay. Other things that are sometimes useful are things called index fossils. Index fossils are used as a reference for dating the rock layers in which the fossils are found. These are fossils of organisms that were very widespread and lived for a relatively short period of time. The layers of rock that they were originally found in have been dated. And it's known that the ages of rock in which you find these particular kinds of fossils are going to be pretty much the same age. So they give you an age range about things. And these are usually things that are kind of microscopic, like diatoms and other kinds of small creatures that live in the ocean that were very widespread for a relatively short period of time. So a little bit about one thing that they use fossils for is determine what kind of environment the organisms lived in. And so if you're looking at, for instance, if you're collecting fossils of an ancient shoreline area, if you find in the upstream area from the shore, you might find some pollen grains that might lead you to think those were pine trees. Okay, you might find an elk skull that would lead you to think that elk probably lived there. So that tells you something about the area there. Closer down to the shore, you would probably find similar pollen grains that wash down, but you'd probably also find clamshells. And then a little bit deeper, you might find clamshells along with shark teeth. And so that would give you an idea that some of those things lived pretty much at the same time. It's just you were living in an area that was changing from terrestrial to aquatic here. And again, as a reminder, when you look at fossils in layers of rock, remember the oldest rocks are going to have more simple fossils. And over time, you may see some that are somewhat related to the ones that were there before, but they're a little bit different in some ways or another, more complex and somewhat different. Here's some examples of ancient creatures looking like modern day creatures, very similar. This is a woolly mammoth. Woolly mammoths were related to elephants. There's some significant differences between woolly mammoths and elephants in that they had different kinds of more robust, I guess, tusks. They had a thick coat of heavy fur. Modern-day elephants have shorter tusks. They're a little bit smaller. They usually live in arid areas that are relatively dry and relatively warm compared to the cold areas that the woolly mammoths lived in. They do have hair, but their hair is much shorter and much more sparse and not nearly as thick as you find in the woolly mammoth. And so there's some similarities, but there are definitely some differences. And you've learned, I'm sure, in middle school how fossils form, just as a quick review. So an animal dies in an area where the landscape changes relatively quickly. So this is a part where there was a flood, perhaps, and the flood covered up the dead animal, which was eventually became covered in deposits of sand and things like that. And over time, more layers were added on top of that, and it became compressed. The flesh rotted away, of course, and the bones became mineralized. In other words, they were infused with the minerals that were in the rock and no longer composed of bone. And then over time, those upper layers may have worn away, and you end up with an exposed skeletal remains on or near the surface. And that's generally what we think about fossils and how they form and where they and how they come to light for us in the present day. Another idea about that supports evolution is biogeography. This is when you find similar fossils in very different locations on Earth. You know that before the present day, there were different configurations of the continents on the surface of Earth. There was at one time a supercontinent called Pangaea, and then Pangaea broke up into a couple of different continents. The southern continent was called Gondwanaland, and it was mostly composed of South America, Africa, India, Antarctica, and Australia. There are various fossil remains of certain dinosaurs, certain ferns, and other kinds of organisms that are found in very different areas, from Central South America all the way to Central Africa, Africa to India to Antarctica, and so forth. And when you find those fossils in those very different places, that gives you some evidence, for one thing, that they existed at the same time in similar habitats in those areas, and for another thing, that the continents have moved. And then we also have homologous body parts. Homologous body parts are parts that are similar in origin. They have different functions. And so here we have the forelimbs of four different animals, 4 different mammals, as is shown here. Notice that the organization of the bones in the forelimb are the same, but their functions are very different, and you can see that the limbs are adapted for how they're going to be used by the organism. Humans generally use their arms to lift and grasp things. We have an opposable thumb that allows us to hold on to things, and so you see the one bone, 2 bones, small bones, longer bones, and digits here. That's the pattern that you see in the forelimb of a human. You see the same pattern of bones in the forelimb of a cat, but the function of the cat's forelimb is very, very different. They're actually walking on their toes, as opposed to walking on their feet, so to speak. In a whale, again, you see the same pattern: 1 bone, 2 bones, small bones, long bones. Okay, and this limb is adapted for swimming in the water, for navigation and movement. And then finally we have a bat, and again we have the same pattern of bones: 1 bone, 2 bones, small bones, long bones. And here we have the limb is adapted for flight. And so homologous body parts are really important. I'm going to attach another video for you to watch about homologous structures that's more entertaining than what I'm showing you. It's from SciShow, and it's Hank Green, and he's really kind of funny to watch and gives some really good examples of homologous body parts. Here we have some pictures of a different picture of some of the same limbs and some different ones, including a frog, and a bird, which are different groups of animals compared to mammals, as well as a horse that shows the fusion of those phalanges to make the hooves. We also see homology in the embryos of organisms. If you look at the embryo stages of various animals, you will see that there's some very strong similarities in what the embryos look like at different stages in the development process. And so there's a section in your worksheet that that discusses this and lets you make some observations about the similarities and differences between the embryos of the different animals. Here we have this. That's not to say that we're identically the same, but we go through similar developmental stages as we develop from an embryo into an adult. Here are a couple of things that you see comparing a chick embryo to a human embryo. There are these pharyngeal pouches or gill slit areas that are similar there. The post-anal tail that humans no longer have. Chickens have a very small one, but they do have a tail. And so there are some definite similarities there in the structural forms that you find during development. There are also similarities in the DNA or in the genes. There are actually these sets of genes that are very highly conserved in all kinds of animals called Hox genes. And these are genes that are developing the location of and the arrangement of body parts. They're not necessarily how to make the body part, but they're when and where to make the body part. For instance, the fruit fly has some genes that control the formation of eyes here. It doesn't say how to make an eye. The fly makes the eye that it knows how to make, but it does make an eye there. And if you take those genes and move these genes someplace else, it'll make an eye in different parts of the body. It'll still make a fly eye, even if you take a gene from a different animal and put it in there, but it'll still make a fly there, make an eye there. And so you see some similarities in all the way from flies to humans and everything in between. So very, very strong similarities in the arrangement of our body parts based on some genes. And a couple of things about vestigial structures in humans, okay? These are, again, vestigial structures are small traces of homologous organs that are no longer useful. Like limb bones in snakes. There's some snakes, when you look at their skeleton, they have these little bones in the latter part of their skeleton that look like the leftover parts of limbs or legs that snakes don't have anymore. Humans have a few of these too. Pica in your eye, this little membrane here in the corner of your eye, is actually the same kind of structure as the nictitating membrane that you see in fish, and that little third eyelid that you see like in cats when they blink their eyes. And so we used to have a similar structure. It's reduced in size because it's no longer necessary, and so now it's a vestigial structure. And that's the end of the notes for this particular part. Be sure and watch the SciShow video for some more interesting things about vestigial structures, and we'll see you the next time. There's also a worksheet on evidence for evolution that you're going to spend a couple of days working on, and please feel free to refer back to the notes if you need to during the period of time you're working on that.